In the meantime, I want to shift gears here in the interest of time and move along to some conversation around analytics and assays. So what are some of the nuances that mRNA therapeutics present in analytical methods testing, and how are those nuances being addressed? Thomas, you're the lucky one who's largest on my screen right now, so I'll start with you on that question.
Yeah, I mean, I think that as far as nuances, you need to still, I mean, we kind of referred to this. I mean, you need to determine what are the critical quality attributes that you want to have for your product, right? And then you need to make sure that you've developed those assays. And you should start to use those assays, during your proof of concept preclinical studies, right? Because you want to make sure that you understand what the quality parameters of the product that's giving you the potency that you're seeing in your preclinical studies, because the goal, of course, is then to bring that into your manufacturing facility. So you want to have those, you want to define that early on, you want to apply it to your proof of concept preclinical studies, you want to tech transfer that to either internal or external manufacturing facilities, and then they should be utilizing some of these assays while they're doing their process development, and you can do some in-process testing to make sure that you are retaining those critical, that you're retaining the attributes of your mRNA LNP complex. So I think that's going to be critical. When we start to talk about therapeutics, I think one of the main things that we're going to see is that, with vaccines, we are actually using relatively lower doses, right? In order to, it's an intramuscular injection. We're using, 50 to 30 to 100 microgram doses in order to stimulate an immune response. And, in that case, when we're going to therapeutics, we're going to be incorporating larger doses and maybe more frequent doses. So what we're really going to start to see is that impurities are going to play a larger role, right? Because now we have larger doses. So with respect to nuances or potential improvements, we're going to have to continue, and I'm saying continue because there's a lot of improvements in the assays that we're using, but we're going to have to continue to optimize and get more sensitive assays that can detect these impurities, because they may play a larger role, especially since some of the impurities could be immunostimulatory, which is not bad for a vaccine application. But when we get into a therapeutic, that's not something that we want to have. So I think the keys are going to be, early defining what the assays or your critical assays that you want to run, and then being aware that we're going to be going with larger doses and to continue to improve the sensitivity of the assays that we're using to look at our residual impurities. I mean, things like your enzyme that you've used, your double-stranded RNA, your cap analogs, residual DNA, et cetera. Those are going to be playing a bigger role with our therapeutics.
Yeah, I'll echo a lot of that, right? The impurities are going to be one of the biggest issues. And in my opinion, especially the lipid impurities, right? So the lipids are already known to be the most toxic part of these formulations. It's not the RNA, it's the lipids. And so those impurities, what are they? Are they toxic? How much can we have? And then moving on to the RNA, right? The integrity of the RNA. What's happening with the tail? Do you have secondary structures? And a lot of this really kind of gets back to what I'll call the core problem, which is the fact that you've encapsulated this RNA. And now to analyze it, you have to deconstruct that lipid nanoparticle. Mm-hmm. How do you deconstruct the lipid nanoparticle without disrupting everything about it? As far as, by deconstructing it, are you creating impurities? Are you actually seeing what was there in the drug product before you deconstruct it? I'll give an example. We actually, in one of our assays, we were deconstructing it, and we found that the reagents we were using to deconstruct the lipid nanoparticle actually were increasing the impurity of the RNA. So we just put the RNA in those same reagents and found that it was decreasing our integrity. So that's a problem. How do we get around that? And I think those are some of the nuances that you don't have to deal with in a lot of typical formulations.
Yeah. So is it a matter of? No, go ahead.
I was just wrapping up. Yeah, go ahead.
Yeah, I was just going to say, is it a matter always of how do we get around that, or is it possibly a matter of how do we avoid it in the first place, or is that?
It's a great question. Yeah, is there something that we can do to avoid having to deconstruct it? Right now, I'd say we don't have the technology or the methodology to do that. Maybe somebody's working on that. I don't know. That would be fantastic. But as of right now, we're still having to find ways to deconstruct it and kind of analyze it piece by piece.